Well, welcome everyone. This is Sue from Jersey Mycological Association. Today is Taxonomy Tuesday. The first Taxonomy Tuesday after our first foray of the year at Stokes Lake. I wouldn't even say it, and I'll just say Lake Oak. If anyone has anything to share, please say so in the chat. Otherwise, we can start with the people who have sent in something. The first person who sent something in was Tom. Tom, I think you're still on.
Yeah, can you hear me?
Yeah, I can hear you. Do you want me to
Okay.
show them for you?
Okay, sure.
Okay, can everybody see this glob?
You want me to explain what I saw, or do you want to explain
Sure.
it? 'Cause,
No, you talk, because you're fine.
well, you know more than me.
Yeah, I know, but this is your
Yeah.
first
I never
slime
saw.
mold, I guess.
Yeah, I never saw these before, and they're basically like puke. They were on chip mulching, and I took 3 pictures. They're very. I like that's the first one. And then like. The second one, there were 2 side by side, so I turned one over and I was surprised. Yeah, that second one,
Yeah, that's the second one.
there's no mycelium. It's very thin. It's just, just wood chips clinging to the bottom. And on the one on the left there, you see there's uh, yeah, that's the third picture. It looks like how it disintegrates. And the second picture, if you show that again, Uh, I scraped my finger along the top of it. You can see towards the right side on the image on the left, and it looked like it was changing color. And the top surface was like a paste, very soft paste, and there's a little more structure underneath that. But so I was puzzled, and I just sent this in. I thought it was a mushroom, but Sue sent me an email saying it's really a slime mold and it's not a mushroom and it's pretty common, but I never saw it before.
Is this your first year with mulch, or is this in your
Oh,
property
this
or
was in
someplace
a
else?
public parking at the train station in West Windsor.
Okay. Yeah. Any other opinions in this being the dog vomit fungus or slime mold, shall we say?
I agree.
Yeah, if you would have got there earlier, it would have been all that yellow color.
Yeah, I noticed when you touch it, it usually comes off on your finger and exposes a black surface underneath.
What? And purchased a piece of land.
I know somebody who eats this.
Oh,
And then vomits.
I don't know. He said it was good. A guy from Western PA Club. I forget his name. Oh, geez, I wish I could remember his name. He's been in that club a long time.
And you always find somebody to eat something.
Okay, but I guess you're eating a live animal that way. Okay.
Well, he cooks it.
Oh, okay, boiled alive. It'll be hard to scrape it off and just get the dog vomit and not the mulch.
Yeah, I imagine it takes a fair amount of preparation. I don't know. I haven't tried it.
What Tom has described is all of the characteristics of that slime mold, like other than seeing it when it's fresh, which is more like the consistency of a uncooked scrambled egg. But yeah, it would eventually turn kind of sooty and gray-black, which is the formation of the spores, its final stage.
Oh, this
well,
one?
you had a bunch of that, and there it is.
Yeah, right. That's nearly a final stage.
Yeah, I definitely have not followed my slime molds when I'm out there. I just said slime mold, move on. But yeah, it's nice to see the life cycle.
I can't tell people enough how fun it is. If you got one of these in your yard and it is turned to that spore mass, shoot it with your hose. It just like the water bounces right off it and it just looks like it's smoking.
And you want to spread it all over your yard? Is that why you're shooting it with a hose?
No, that's just for fun.
Okay.
I didn't notice any smell.
Yeah. It doesn't have a smell.
Yeah, I don't think it's got a distinct one.
So luckily you have slime molds and not stinkhorns in your mulch, and you would have a smell.
Good fierce.
Okay, anything else to say, Tom?
Oh, that's good. Thank you. Thanks for the comments.
Um, Okay, Brandon, you want to go next? Then Lila. Let me stop my share.
Yeah, I can go real quick.
Don't have to go quick. It's early in the evening.
I know. So I found a slime mold this week as well. I'm pretty sure it's Hemitrichia. I am struggling. Well, there's two things that it could be. I think it's clavata is one of them, and
I don't see a picture yet. You're still working on that.
oh, my bad. Got me?
Yes.
All right, cool. So this is the thing that I found. Genus Hemitrichia, and found it at a cool stage. I mean, it's the capillation is coming out of the ice cream cone. Different photos, you can kind of make out different characteristics. You can barely see the black stalk, and my stereoscope washes out color. So this was more of a true color. They were yellow. They did dry a little bit darker. But here you can see the feet, and they're kind of grown individually, not in clusters, but there were a lot of them.
Does this one tend to hang upside down instead of sticking up?
It does both. Most of them, I noticed the ones that were pointed up, but then as I turned the this was a stick really. There were some on the underside. They were both on the bark and on the the parts where the bark was removed already. This is a cool shot.
Is this one of the things that in the earlier stage it looks like a stalked fish egg?
Yeah. Yep. The orange. There's an orange one, and they call it, I think, salmon egg or something like that. And they really do. They look like fish eggs before this bursts open and all the capillation comes out.
Yeah, I was gonna say, is it something that bursts open or one that you kind of like blow out like a
Well, here's enough.
like one of those tube slimes or whatever?
No, it bursts. It bursts open. I mean, this one, it's really throwing that capillation out there, and all the spores are in the capillation. So then, wind or whatever blows them away.
That thing's awesome. How big are they?
A couple of millimeters, not the tiniest thing I found this day. Not the tiniest thing I found on this stick, actually. Yeah, probably stood 2-3 millimeters.
Now, which color was it? I just saw yellow, and before they were all, orangey red kind of thing.
Yep, like I said, my stereoscope kind of washes out color, and this is the true color. They were more
Okay. Yeah.
more yellow, and then as they dried also they kind of turned more dark yellow, and that and the combination of my scope kind of washing out color. Yeah, the true color was yellow in the field. This is the capillation, and I didn't have the best luck with microscopy. The real way you tell the difference between the two species that I mentioned in the beginning, which are clavata and calyculata. They're super, super similar. And you gotta look at the spores at 1000x, and I just didn't.
Is it because of ornamentation on the spores?
Yeah. I saw some ornamentation, not that I could really. It didn't look like, it should at 1000x, obviously, for either species. So I wasn't able to really capture it all that well on the phone or in the photo. But the capillation cool.
Okay. Let me see here. Calyculata has slightly smaller spores. And clavata, usually the base is more tapered rather than a thin stalk.
Kind of like, can you see the base here?
Yeah, sometimes it's hard to tell the difference.
Yeah. That's what I've kind of I think I got the best that I could do without going to 1000x under the scope to look at the spores, and I was like, I'm pretty sure it's one of the two, and
yes, it probably, I'm sure it is. Calyculata has more of a reticulated spore, slightly smaller than the other one.
Yeah, the only other thing, maybe the only useful thing, and I had to kind of trick my camera into capturing what looked like a little bit of a thick cell wall, which I think one of them, maybe the spores have and the other one doesn't. But I think I just kind of doctored that photo a little bit by messing with the focus. So yeah, one of the two, but I think they're really cool.
That does look like a thick spore wall.
Yeah,
for sure.
But you know how sometimes when you get something just right out of focus, it kind of
it looks
Yeah.
thicker.
Actually, because when you're looking at something like a spore, and the spore itself, occupies many different levels, like lots of different planes that pass through it, perpendicular to your line of vision. And when you're playing with the focus, you're just changing which plane you're looking at. And sometimes one plane is better than another, to see how thick the walls are. But other features using that same level of focus get blurry.
Right.
You usually have to focus up and down to see the reticulation.
Right.
Gradually focus up and down and just get an idea of the reticulation.
And on these ones in particular, if you've ID'd them before, Virginia, if I believe the spores are pretty small, right? And do you look at 1000x, or are you able to see that?
I always look at 1000x if I'm looking at spores.
Gotcha. Yeah.
You can't really, unless they're large or really distinct, you can't tell anything until you look at under, at 1000x.
Right, because I'm used to working like the Scrutilinea that I found last week. You can see the reticulation at 400 because the spores are like 20, 22 microns.
Well, these are somewhere like 6 up to maybe 9.
Right. All right, cool. Well, I'm okay with it being one of the two. And then I'll kind of just go through this one quick because I'm pretty sure it's Orbilia. It's at least in Orbiliomycetes. Just one of maybe the closely related sister genera.
All those things.
But this was on the same stick as the Hemitrichia we just looked at.
Orbilia is Aspen.
And I said we'll go through quick. You can see we got a little bit of a lip to it. These little discomycetes, really, really small, anywhere between 0.75 millimeters. I think the biggest one that I found was about 1.5 millimeters. And if anyone has an opinion on this stick, maybe what kind of wood it is, go ahead and give me some guesses. My best guess is it was right under a standing birch, dead birch. So it was in the mud. I mean, in a creek, pretty much. But the only cool thing, the only success story here is I finally got spores out of an Orbiliomycetes. These things are super hard to work with. They go xeric really quickly, which means that like when they dry out, their cell walls actually die, which is pretty interesting. So if you don't get them home kind of hydrated, they don't really like to drop spores. So and also everything is hyaline. Kind of similar with looking at the spores in the slime mold we just looked at. You'll notice these spores are pretty blurry. That's because without getting them out of focus, they're pretty much see-through. And you can see they're also quite small. This is at 400x.
Do you use any? You use some stains, right?
I do, but I always like to start with water. And with these things, they're so small, I just don't have any more material to mount.
Yeah, you get one shot and that's it.
Yeah, and I like to do it in the water to get the the colors of things. And while stains they do highlight certain things under the microscope, they can also kind of wash some things out, I think.
I start with water as well, and then I'll stain
that same
that
material.
exact same amount.
Yeah, I've done that. But yeah, I probably should have done that. Would be better practice. And maybe it takes some imagination to see these pretty much almost invisible paraphyses. But you can see they're long and slender with like a subcapitate thing going on. And they don't have a central digit. Now, I don't know. Do you guys see what I'm seeing there? Or do you have to take my word for it?
It's better further out. Yeah.
Oh, yeah, kind of. You're right.
What about those wider things? Are those the assay?
They are correct. Yep. And they are. I've said this before. The only other time I found Orbilia, they're kind of baseball bat shaped. They kind of have this like abrupt end, and they taper down, and then they have a little cleft foot. Looks like the end of a baseball bat. But yeah, that's
stuff like that too is real nice with the dark field.
Yeah, I still haven't mastered that. Back to this stick. Anyone opinion? What kind of tree it could be? Yeah.
Can we tell by the leaves? Are those leaves?
No, this was. I found this stick in a creek, pretty much.
Okay.
And got it up out of there so that I wasn't taking pictures in a creek.
Does it have does the little pieces of bark have any like horizontal markings? Because a lot of times birch bark will have that.
No, I didn't really see it.
Yeah,
but it was also a stick.
Yeah, stick might not look the same as the bark.
This
on
is
a trunk.
this is one of the bigger Orbilia that I found. So that's maximum 1.5 millimeters. Cool. But yeah, a good practice, especially with Orbilia, the different species are pretty host specific. So I found this one today, and I think I got a pretty good idea that it's this Guepinopsis buccinia. Tough to photo. These things are small too. Little goblets or cups on a little foot. Stipitate. This color is very true. They did have a bit of a translucent quality to them. They were rubbery, kind of like jelly. Weren't real fragile.
This photo is pretty good. You can see the concave
Yeah.
upper part. So
And
that's
then-
that's a good way to know you probably have what you have and not, some like Dacryoponax.
Right. And then another thing, Dave, right, that you want to look at for this is the kind of veining or ribbing on the stipe.
Okay, yeah, I forgot about that, but that sounds right. But I've confused these with Dacryoponax a couple of times.
So these are a jelly fungus, not an asco.
Correct. Yeah, these are basidiomycetes. If the idea is correct.
Yeah, a lot of times they're a little more orange than these, but the shape seems to be right on.
Yeah, and I double checked with a couple sources, but only briefly because I didn't remember that the stipe and the underside of the cup was ribbed. I thought it was like a pseudo gill or something. That's how I remembered it, but I checked. I think it was Myco Quebec has this thing, and they have a couple pictures that look almost exactly like mine, even down to the color. So cool.
Nice.
Do I want to share anything else down there? All right, let's at least get me back to school on some Amanitas here. Yeah, I'm calling this Validae. I don't think it's section Amanita, but am I totally misreading that? You can see the collar here has got a little bit of yellowing. Your collar to the bulb. Marginally striate, some UV remnants that like pale yellow, off-white, pale yellow, yellow ring. This thing was pretty small. Do I have the section correct at least?
The section is correct.
Yeah, I agree.
The marginal striations are there probably because it's drying out. Validae should not have marginal striations, but there are exceptions.
I find Validae, Flavoconia, and some of the rubescent ones occasionally have short striations like this. But
I would call this one elongata.
Elongata, or Flavoconia.
I wanted to call it elongata, but I'm not good enough with these things.
Well, supposedly you can. This elongata, the name actually, if not totally, partly comes from the fact that the spores have a larger Q value than Flavoconia. But it's pretty close though anyway.
Oh,
so
okay.
Yeah, but I
I
think
just kind
that,
of
But
Go
but
ahead.
then Flavaconia itself is a species complex. So
yeah, yeah.
All right, I think I'll probably call this one elongata.
yeah, propose elongata, and I doubt that anybody's going to argue with you.
Yeah.
Yeah, I wouldn't argue with that.
All right,
Yeah,
cool.
the stipe is really white and thin. That's a
Yeah, stipe
really
is
cool.
white. That's important. Flavaconias typically have yellow stipes,
Okay, cool. Nice. All right,
and
great.
the color here is more yellow on the cap on the disc, and than orange. You know, it's more of a kind of a uniform yellow. Maybe the edges are lighter, but I mean it's arguable. But you know, it could be. I would go for elongata just because, if it's a real species to begin with.
Right. Okay. Nice. Put a name to a thing. All right, that'll do it for me. Thanks, everyone.
Okay, thank you, Brandon. Next up is Lila. Okay, let's see. Share. All right. Start with this one, this Helvella macropus, which I thought was really more of a spring item. I found this up north in New York State, and basically discovered it by accident. I was not expecting to find anything right where I was walking. I thought it was pretty cool, very ornamented, if you will, on the underside and along the stipe. This one appeared to have some sort of mold or hypomyces. I pulled it up. You could see it was pretty well attached to the soil. This is in brighter light. It's kind of tan on the outside. And I think this is, they tend to start splitting apart as they mature. And this is another one that I think was infected with something. And this is an oak associate? Possibly. I can't remember now. Let me see what I wrote about my notes. It was a mixed forest, hardwoods and conifer both present. The weather had been, crazy up there, like down here, wet, then warm, wet, then very hot for several days. Until the time I found this, it was about, the high was like 60. So it may have come up in the cooler weather. I thought that was interesting. It's the first time that I recall actually finding this. I don't know if anybody has any other thoughts on it.
I would have to look up the features, but you did.
Mhm.
So, but I would just point out that some of the things you mentioned, like the underside looking like sandpaper, being like having a lot of like scales or ornamentation, and also the stipe also having stuff on it. Those are some of the things that help separate several of the species in Helvella that are basically these little stock cups, and that are gray or yellow.
Yeah, you should check out Myco Quebec. They have, I would, no argument here for macropus on this one, but there are a couple species that they are calling a group of difficult to distinguish species that have cup-shaped or disc-shaped or spreading apothecia, finely pubescent with a thin stem and no prominent ribs. So looks like 4 or 5 species they have. Just the difference is about.
The macropus, mushroom expert says is unique in having spindle-shaped spores.
Yeah, I was going to say that spore morphology is helpful with these.
Yeah, I wasn't set up to do any micro work there, so and I didn't save this, I don't think.
Yeah, you know what? If you,
Actually I did. It is in a fridge in a bag, but
you probably won't drop spores now, though.
no.
Well, I mean, if you really want to, I mean, it's up to you how much you want to try to remember to do. But one thing you can do is carry microscope slides with you, and then if you find something really interesting, just try to get some spores to drop on a slide,
Right.
and then wrap it up in wax paper. And wax paper is pretty good. It's non-reactive. And just label, put a little label inside the wrapping, and then when you get home, you can look.
Yeah. All right. What else? Susan's on. I was going to go back and look at this, but I forgot about it, so I didn't pull one up. But maybe she can recognize it from the top. Some sort of Hydnellum. Under hardwoods, mostly hickory, oak and maple present, as is one hornbeam.
You didn't pull one up, you said, right?
No, I didn't. But
Oh, but to have it from the side.
yeah, it's
It's not really, well, yeah, it looks like it's going to be one of the what Americans call Hydnellum pinticola or ferruginium is what it's been decided. It's equal to the European species, but that's just a guess, and that's because of the thicker stem. The spines and things have not developed enough, and also that thick lip kind of cap makes me think of that one, and the pinkish kind of brown color.
Yeah.
But that's really way too hard. It's got to be a little more mature to even take a good guess.
Yeah, that's why I left it. I figured it was too small to really do anything with, and now I'm seeing it.
Actually, looking at the top, it looks a little bit like a true scrobiculatum, but again, that's kind of tough to tell. But if you can go back where it is some other time, might be a good dyer.
I know, that's what I thought.
And when it's mature, if you can leave it till it's, really brown and old, which will at least be several weeks.
Okay. All right. Yeah. So this was like last week sometime. So I'll go look later this week.
Is this up in your place in New York?
No, this is in Jersey. This one.
Oh, really?
Yep.
Wow.
Yep.
That makes me think more scrobiculatum.
Okay.
What color do you get out of the?
For the dye?
Yeah.
Tealy blue-green colors.
Really?
Yeah. Yeah, and what's important is pH, adding ammonia to the bath as it's cooking. And you're mordanting your yarn wool with iron, not iron, alum or iron, either one.
All right. I will keep an eye on that.
Mm-hmm.
What else?
Oh, interesting bolete.
So, all right, my notes here, about a dozen. So these were in New York, about a dozen scattered under oaks, probably within the distance of the height of a hemlock that was in the nearby woods. They're growing in a lightly used farm road between two fields. Fields are separated by a stone wall along which several oaks grow, the road following alongside. So possibly Neoboletus
Piece. Can you make it bigger? Yeah.
Yeah.
Yeah, see, that's still kind of small for me to see. Yeah, that's looking what I would call Boletus subvolutipes, which has now got a new genus, right, Igor?
Yeah, it's a Neoboletus, except that I don't think this is a Velutipes.
No?
The cap color is wrong.
Yeah, it's true.
I think this is subluridellus. This is the oak-associated sister taxon to Subvelutipes, aka the chameleon. And that's pretty sure it is the chameleon. Not sure what, Dave has a lot of experience with Subvelutipes. So
I have a hard time telling in the field the difference between the chameleon and the true Subvelutipes, but this looks more like the chameleon. Look at the stipe is really, it doesn't have much going on. It's just
Subvelutipes always has red, and it's flushed like in flames in these long reaching streaks. It's not punctate. The cap color really to me suggests the chameleon.
Yeah, the cap color is kind of maroon, sort of.
Yeah, kind of a dark fox red color.
Yeah.
So yeah.
Is that going to be an official name, the chameleon?
Well, I call it the chameleon because the true identity of that bolete. We got type specimens by Smith sequence, so this is going to be.
Yeah, I just see it in quotes, and I'm on the online. I'm just wondering if that's a real
proposed
name or not.
I call it Neoboletus chameleon, which is a noun in apposition.
Okay.
Neoboletus the chameleon. So it's a provisional name, and that's how I've been calling these things because we didn't know the identity of Smith's subluridellus till recently. So now that we have the type specimen sequenced, unofficially, we can call it subluridellus now.
Okay, so we can start calling it that now?
Well, I mean, I wouldn't call it on MO, but, in our meetings we can, yeah.
All right, okay.
Could you put that in the chat? I'm not quite spelling it right.
Yeah, we need to wait for that paper to come out, which is going to be later in the year, by the Dentinger lab. Which name?
The one that's not the chameleon.
What, Subvelutipes?
Yeah.
Subluridellus, I think you said, right?
Well, no, I said chameleon is Subluridellus. Subvelutipes is a sister species. So
Right.
Which one do you want?
I'm confused. Which one is the one we're looking at?
We're looking at what will become Neoboletus subluridellus, I think.
Okay. That's the one I want.
Okay, subluridellus.
But it is, Lila pointed out that these were growing near oak, but there was hemlock not too far away. So that does make things a little bit confusing. But these do look more like the subluridellus.
But in my experience, whenever we've seen the chameleon in the woods at Stokes, a different location, Kittle Field. They were growing under oaks, and there were hemlocks nearby. But I'm pretty sure it was the chameleon, as far as you could tell just from the morphology. The chameleon has the punctae. Okay, the Subvelutipes doesn't. Its red color is more like the background that's solid. It's not consisting of puncta or squamules. And then the cap color to me suggests strongly subluridellus. It's a highly, highly variable species, and it does mimic Subvelutipes quite a bit in some ways.
Yeah, well, in fact, there's more than a few field guides out there that lump together this with Subvelutipes. And in fact, there are several field guides which describe Subvelutipes as being an oak associate that sometimes is found with conifers.
Right.
So those reports we believe those reports are just wrong.
Correct, they're wrong. The main difference between Subvelutipes and subluridellus is going to be the ecology.
Yeah, there's some subtle microscopic difference. Subvelutipes has a little bit wider and longer spores.
Right, but you've got to measure them, and you've got to be very thorough in making sure.
But it's just to be able to provide a little bit more support, for. But the DNA, I guess Subvelutipes has a really consistent ITS.
Oh yeah, subvolutipes, the variability in ITS and subvolutipes is non-existent essentially. I think I've seen
That's
only
what
one
I'm saying.
collection
Yeah.
that was one base pair off. The rest were completely identical to each other from different parts of the country.
Mhm.
Subluridellus is going to have variable ITS. You know, whatever the genetic distance is, 0.5% or 1%, I forget. I have to look it up, but it's going to be more variable, just because it's more geographically widespread.
Okay, moving on to parasols.
Yeah, these don't get any respect, but I thought I'd show one because couple of these because I played with them on the microscope this morning. So after the rain, such a fragile stipe. And some nice spores.
So is this at 400?
Yes.
Yeah, so so each division is 2.5 microns, I guess.
Yes.
Yeah. So these spores are kind of close to 10, right around 10. Looks
Yeah,
like
a little bit more.
a little bit more than 10 here and there. Yeah.
Yeah,
Well, the
12
length.
to 13 by 8 to 9.
Oh, is that what you got? 12 to 13?
Yeah.
Oh, okay. Yeah, just glancing, they looked like maybe more like 9 to 11, but you measured them, so yeah.
I did.
Yeah, that's.
Yeah, there were some that were smaller. I probably gravitated more to the larger end of it.
Yeah, well, I've been told by, professional mycologists to not do that.
Yeah.
But honestly, when you're looking at 400, you kind of have to be wary of the smaller ones just looking smaller than they are because of the way they're positioned.
Yep.
So I do tend to gravitate more toward the maximum length.
Well, that's
So
why
the-
you need to do that 1,000. So you find your smallest spore and then your largest spore, and then you randomly measure the remaining 18 spores. That's
Yeah, I know.
5th and 95th percentile, and then everything in between.
And just for contrast, I decided to look at spores for the Pseudocolus fusiformis for fun. So we'll jump to that, and you can see these things are tiny in comparison.
Wow, you're brave. You got spores from one of those things?
Well, I just smashed the smelly stuff that was still left there on a slide and
yuck.
Took a look at it. So they're tiny, 5 by 2 basically on that order. So
And was that informative?
not really, because I knew what it was. I didn't really need it, but I was curious to see what they looked like and see if I could get them out of that goo. And it was easy enough to do.
Unimpressive looking.
Yeah. But anyway, I had fun, so that's enough, I guess.
Okay, yeah, we've got about 5 people to go. Chris, you're next.
Awesome. This was up at Stokes. I'm calling it Antrodia. I got the Club Polypore book out, and it said it should have angular pores, which these seem to be. But when I got home and I took a little chunk with me, it didn't drop any spores. But once I started cutting little chunks off, there were plenty around to look at. And then when I started looking at spores from different Antrodia online, those all seem to be much longer than wide, and they all seem to have like a pretty distinct apiculus.
What's giving the green color?
I have no idea. Just they're just mounted in water, and it's just the light of the microscope. But yeah, they definitely do have a color to that gutule.
I can't speak to finding spores at something like this. I never seem to get a spore drop from a polypore like that. But I always call these things Antrodia. The angled pores that are kind of layered like that. Whether that's right or not, I'm not sure. But I think you're right. I don't think those look like Antrodia spores.
Yeah, I mean, they seem way off. So I have no idea what this thing is. I don't think.
You know, I seem to remember there being something that like Neoantrodia or something like that, but I just looked and I couldn't find anything. But I mean, they certainly look like what Antrodia, like Antrodia looks kind of like, you know, pipe organ upside down or something like that.
Yeah.
Yeah. What is that? What did you say? Neo.
I forget. It may have been
Antrodiella?
Antrodia, something like that. It like had Antrodia in it, and it looked like Antrodia. You know, I ran across it a few years ago trying to identify something like this, but I just looked on Michael Quebec. I did a search just on the letters in Antrodia, and I didn't get anything.
The other thing I was thinking is like, I mean, is there a possibility, because this thing was soft when I collected it. The entire log the stump that it was coating this stuff was all soft. And I'm just wondering if maybe they weren't mature, because it didn't drop any. I found them after I cut some pieces and mounted it.
Yeah, that's kind of always the question. Also, when I find something like this, I don't know. These things are tough, in my opinion.
Yeah, I figured, you know, what the heck, I'll take a swing at it, you know. But that's all I had to share. Everyone did find lots of nice stuff up there now.
Thank you, Chris.
Thank you.
Jason, you have something? Even though we
Yeah,
didn't
can
see
you
you
hear
at the
me?
foray?
Yeah, I couldn't make it. All right, so this is some sort of fungal pathogen on a rare orchid called Isotria medialoides, which is the small world pogonia, which is federally threatened and state endangered in New Jersey. I was doing a survey of this last week and found one plant that looked like it was infected with some sort of pathogen. I'm guessing this black area around the leaf is kind of the necrotic tissue, and then these light brown circular areas are the fruiting areas, fruiting bodies. That's my assumption, although I'm not completely sure of that. So I collected a leaf and I tried to cut out one of these light brown circles to see where I can get under the microscope. And I got some things that kind of look like spores. Here's one. It's not the best photo. Here's a better shot. At first, I thought they were muriform spores, but now I'm thinking they might just be stacks of one septate spores.
Well, what does muriform mean?
Like the brick shape, wall shape. And they have like the divisions.
Right. Yeah. So, and you said you're now thinking that these are, yeah, that would still be, septate vertically and horizontally, right?
And horizontally. So, I mean, this one looks like it might have. It's hard to tell. I think I have a better photo. I mean, this looks like just one septate spore down here, these guys.
Well, some of these crazy, ascospores that are septate multiple times in multiple directions, they get those septations with maturity. You know what I mean? So,
okay.
Immature ones might be less septate, and then the larger mature spores, develop.
Right.
You said this was on some kind of orchid?
Yeah, it's on the small world Begonia.
So is this considered a rust? Is this a rust or something else?
Well, I'm not sure what it is, but my assumption is that it's a rust. It infected all of the leaves, and then it's kind of extending down the stem here.
Well, I don't think it would be a rust at that point. A rust doesn't want to kill its host, and I mean, it looks like whatever this thing has killed the orchid, right?
Are leaf spots considered rusts?
No, they're considered
something that would be.
They're considered pathogens, right?
Yeah.
Rusts aren't necessarily pathogens.
If this is a muriform spore that's a little bit more mature, I was thinking maybe alternaria. Which is a leaf spot-like thing that has muriform spores. Let's see what the classification for it is. Those spores. And pathogen. Videomycetes.
Spores don't look too bad for that one. Even down to the variable septation on the spores that we were just looking at for this thing.
Yeah. So I'm going to continue to try to work with it, see if I can get some more spores out of this thing. But
did you find healthy orchids?
Yeah, it was the only one that was infected with this fungus.
Cool.
So that's this guy. Then I found a muscicola parasite, a moss parasite, Eocronartium muscicola. You can see it, fruity body coming out of the moss. This was on a rock in a rock wall up in Sussex County. Here's the fruiting structure.
You said it was a fungus on a moss, so is that the moss that has been turned white by a fungus, or is that something growing out of the moss?
No, it's something growing. It's parasitizing the moss, so it's growing out of the moss. Don't know if this is related or not. That's the only thing I was able to get out of it.
Can we see the in situ again?
Yeah,
Yeah, what is that on a rock?
it's on
Is
a
that
rock.
that multiclavulina?
I don't think so.
No.
Maybe it, I mean, I guess it could be, but Jason, you know that thing pretty well, right?
Multiclavulina?
Yeah, the mucosis, right? Is that what you're thinking?
Oh, yeah. No,
Yeah.
that's on rotting wood.
Yeah. Like algae covered wood almost. Yeah.
Yeah. This is definitely attached directly to the moss.
It's interesting because I've seen something not exactly like that, but white things coming off the moss. And I just thought it was some sort of stage the moss went through, like a fruit body kind of a thing. Now I've got to go look at it closer.
Yeah, some of these pictures.
Wow.
Whoa.
Yeah, that's
Yeah, I would probably assume the same thing if I saw that, Lila. No, because is that what you were talking about, that last photo, Lila? The
Which
brown,
one?
the brown things.
So the brown things are
No, it wasn't that. It was
structures.
definitely white things coming off the ends of the green tips of the moss kind of deal, which seemed odd. It's like I was like, wow, I haven't seen any kind of a fruiting thing that looked like that. But yeah,
I bet there's some interesting micro to be done right where that moss meets that structure.
yeah,
Because they're coming right out of that.
yeah. That's kind of what it
That's
was looking
crazy.
like.
Yeah, that is pretty cool.
Yeah.
But also, some of these photos do, I think less so, Jason, your observation, but some of these photos don't look dissimilar from multiclavula mucosis. Just kind of as the structure is, not necessarily where it is.
I think it's
Yeah.
multiclavula mucida.
Oh,
But yeah, I still am leaning towards that personally.
Yeah, this is definitely only something that grows on wet, rotting wood.
wasn't it
Never on rock.
considered a lichenized basidiomycete?
Yeah. Yeah, see how it's got a green base. It's got that green stain. That's the lichen. And these are just the fruiting structures.
Okay.
But yeah, in terms of like what the actual fruiting body looks like, certainly doesn't look dissimilar.
Yeah.
But yeah, I think the thing Jason has is different.
Yeah, this is the algae in the cortex at the base. So that was cool. Here's a bolete. I think it's just Xanthoconium affine.
Yep.
The velvety cat.
Could be purpureum.
No, this is affine.
How do you know for sure?
Oh, it's brown.
Yeah, but the cap looks kind of purple to me, and the stalk looks kind of purple.
No, it's brown. It's the sun. This is brown.
Yeah, I think it was brown.
Yeah, you see.
Okay, yeah, the
it's
stipe
kind of
looks more
brown. Yeah,
brown than-
yeah. It's
All right.
way too brown for purpureum.
And when you were saying purpureum, is that also Xanthoconium, or are you talking?
Correct.
Yeah.
Okay.
Yeah.
Cool.
Now, do both of the species have the blue-green reaction to ammonia on the cap?
Oh, I haven't tried chemicals in these things. I just
Well, I
can
think,
separate
I
them
know
without that.
purpureum, you get a really strong reaction with ammonia on the cap. And I think it's kind of fleeting, but it's a flash, like a bright,
Yeah, I know what you mean.
blue-green
I
flash.
haven't tried it. Typically purpureum, this type it doesn't have any brown. It has these vinaceous or purplish streaks. Sometimes very pale, but it's never brown like that.
Yeah, no, I agree. Yeah, this
So,
type is too brown.
where was this found again?
This was up in northern Sussex County.
Montague?
Yeah, not far from Montague.
Not so very close to Stokes. Yeah.
Yeah.
Yeah, it was very common at the Stokes we found a lot of Xanthoconium athene in there. Tons of it. I would say over a dozen fruiting bodies.
Yeah, I found it in Pennsylvania recently, also in Ricketts Glen. I'm sorry, Hickory Run. But most of them were parasitized by hypomyces.
Yeah, we found one or two fruiting bodies that were parasitized by hypomyces or beginning stages, and one fruiting body was completely covered by it.
So I'm
Yeah, these
guessing you guys
notorious
also
for
saw
that.
a lot of Cortinarius corrugatus up at Stokes.
Yep.
Yeah, that was found
at
at
least one
Stokes.
collection there too. Mm-hmm.
Kind of a musty smell. And the last one I got is this is a caterpillar of the spongy moth. And apparently when you see a lot of dead caterpillars stuck to the trunk of a tree, it's likely that it was killed by a biocontrol agent that was introduced from its native range called Entomophaga mimiga, which is a fungal pathogen that attacks the caterpillar and then causes them to cling to the bark. Eventually they fall to the ground, but if anybody's interested, there should be a way to try to get conidiospores out of the dead caterpillars. So definitely spongy moth, and I'm assuming it was killed by that fungal pathogen, which is apparently just in the ambient environment. It was introduced decades ago, and it's still an effective control agent. So,
That's pretty cool.
That's pretty interesting.
yeah.
And it never produces any sort of fruiting body, or anything. It just kind of.
Yeah, I think it mostly reproduces by conidiospores. I've read that some land managers will collect these dead caterpillars and move them to other areas and really, just kind of stick them in places so that the conidiospores can be released to newer areas. But yeah, that's apparently it. I've seen trees with just hundreds of these dead spongy moth caterpillars.
I've seen that same thing, Jason. So it's cool to know why.
Yep. It's probably the Entomophaga mimiga.
And what about the status of the trees? Are they totally defoliated or only partially because of the work of this fungus?
So I usually see this in the early summer. Trying to think. I would imagine that any defoliation would happen kind of later in the season. So presumably it's preventing that defoliation from occurring by the non-native spongy moth.
Actually, I think that the spongy moths, when in an area where there's a lot of them, you'll see a lot of the defoliation like around now.
Yeah.
And and then by the middle of July, the trees are bare, and sometimes they refoliate.
Yeah. Usually I see these dead caterpillars starting in like May or June. I think it's I think the entomophaga is most effective with wet springs. I think I read that.
So maybe it's helping to reduce the pressure of them.
Yeah.
Yeah.
Is the spongy moth this newly renamed?
Gypsy moth.
Yeah, there you go.
Yeah.
I'm
Yeah,
glad somebody else asked that question because I was just trying to spell it and look it up. Okay, so this used to be called the gypsy moth, right?
Right.
Could somebody spell that spongio thing?
SPONGY. And that's all I got.
Hey, thank you, Jason. If anybody else has joined the call, wants to have something to share, put your name in the chat if you didn't already send an email with the pictures or links. So we turn to the emails right now. Igor, you hardly ever send anything in. So would you like to go next?
I hardly say anything. Well, I do mostly in summertime and the fall. In the spring, probably, yeah. Yeah, I could go next, but can I share something quickly before we go into my emails?
Sure.
Yeah, I want to share an observation of Neoboletus chameleon, aka subluridellus, that looks like lilas. This has been sequenced, not this collection, but another collection that came from, this collection. Let's see, can I remove that? No, I guess I can't. So can you see this one? 427,960.
Yes.
The cap color, the stipe, looks very similar to lilas. Now, this was collected in New Jersey, and you can see the squamules on the stipe, which give it reddish squamules on the yellow background. So this is your chameleon. The observation from the same location that was collected 3 years prior is this one. Can you see it?
Yes.
So so this one was sequenced, and here actually I give analysis of multiple ITS sequences where the differences between the sequences come. So the Y's are the letter Y stands for where the base is either C or T. So you have two basically copies of ITS. One has a C, another has a T, because when you do sequencing, they come together in a chromatogram. So you sequence mixtures. So you have to separate them, and the only way to separate them is to do nanopore sequencing or cloning. So you can see the same appearance. The sort of fluorescent reddish brown cap looks exactly the same as lilas. And this one again is genetically is subluridellus. It's not Subluridellus. And nearby there was growing older fruiting bodies where the caps are more discolored. They look more like something you would call maybe vermiculosoides, just by the look of the cap. They're yellow brown like that. So compare the colors like this, and when it gets older, it turns into that. So very variable. And next to it was growing something else. Maybe somebody can ID it for me. All right, so I'm going to stop sharing and just, again, more credence to my theory that Lila's collection is most likely subluridellus, not Subluridellus.
Igor, where you say Somerset County, is that our location again?
That's the location, yes.
Huh. Interesting.
Yeah, I mean, I've been going there since 2011.
Yeah, since I told you about it, yeah.
Right, yeah. I mean, you told me about this, I think, maybe a little bit earlier, but I didn't start going there till 2011. So when you do a deeper search on the Mo with my name and Somerset County as the location, all observations there that are mine came from that location. So there's going to be over 100. So there's probably there's tons of species there. Boletes, Amanitas, other things.
Oh, I know.
Yeah,
I know.
yeah. I mean, it's a wonderful location. I mean, you can.
Well, I'm glad you're making use of it. That's all, because I
Yeah,
can't.
I'm just documenting all the species I'm finding in there. So it's like my own project, kind of behind the scenes.
Great. Great.
All right, so I'm going to stop sharing so we can go into my emails.
Okay. Okay, is the first one.
Okay, so we found a number of boletes at Stokes. I think we had under 10 species. I think I could recall maybe 7 overall. Maybe there was another one. I have to basically mention them out loud, and maybe Dave can recall something that I missed. So this group of mushrooms that features yellow bluing pores, red caps, and yellow stipes flushed with red, variably, is probably one of the most difficult sections in the Boletaceae because they come from a number of genera. Okay, so this one that has sort of brick red cap and a very mottled stipe, mottled red over yellow background, is a very typical appearance of Biorangia bicolor, and this is Biorangia bicolor. And when you encounter this bolete enough times, and somebody basically points out to you that this is the true Biorangia bicolor, you will be able to discern it from everything else. There are telltale signs for this species. So just remember this image, and
Okay.
the next time you encounter something like Lanmaua pallidorosea, you will see the differences between Lanmaua pallidorosea and Biorangia bicolor.
Okay, this photo helps because the last one I was like, that seems pretty dark, the blue bruising, but this is less so. Is that correct?
Yeah, they do bruise, and they do bruise on the surface of the stipe. I didn't cut these because I wanted to have these intact at the foray so people can look at them. I did cut them at home because I did preserve. Actually, no, I didn't preserve this collection. This one was taken by somebody who wanted to eat them. So I don't have this collection. And I didn't cut it. But yes, it's going to be bluing on the pores and on the stipe when you touch it. But when you cut it, there will be hardly any bluing on the flesh. Sometimes no bluing at all. And
Yeah,
here you can see the cat.
To me, that's one of the main differences between Biorangia and Lanmaua.
Yeah, but
is the
Lanmaua
Lanmaua-
pallidorosea doesn't stain either very often, especially the older fruiting bodies.
Yeah, but when you cut them, you get some sort of more organized bluing inside on the flesh.
In other species of the Lanmaua, yes, but not in pallidorosea.
Not in not in pallidorosea. Now, sometimes I find that Biorangia is kind of variable in terms of the bruising on the exterior. Sometimes if you rub the cap, it'll turn a little bit black.
Right. I guess it can happen on the surface. There's also another species of the Lanmaua that's going to be described as brand new, and the authors are going to keep my provisional name for it. It's called Thalax, and Thalax refers to the fact that it's deceiving. It's deceptive. Separating that from pallidorosea is a little bit challenging because it also smells a little bit like bullion. chicken or beef bouillon when you cut it. But anyway, so Thallax and Polyderosia aside, I think I do have an observation or two on a mow where I put Polyderosia and bicolor next to each other, and they look very, very close. Okay, we can move on to the next one.
Can I just ask one more question about the Lemmaoa Baurangia kind of distinction? Can Lemmaoa spores have that, in my opinion, like kind of decurrent connection with the pore surface and the stipe?
Bicolor does that.
Right. Does the Lemmaoa do that as well, or is that a good way that you can kind of just see that?
Mostly not, but occasionally it can. I think I may have an observation on the mow that shows younger fruiting bodies that do have a kind of a seamless transition.
Okay.
But remember, bicolor is usually more deeply darker colored. Polyderosia, as the name suggests, is powdered rose. It's going to be a different shade.
Yeah.
And they do fade away. And of course, the odor, you will never be able to confuse those two species if you smell them. Now, when you smell this one, it's going to smell like curry.
Yeah.
And that was my working hypothesis for Boletus sensibilis. Unfortunately, sensibilis will remain a mystery after the whole genus is unraveled because the type collection has not been sequenced, unfortunately. So now this species is going to be described as brand new, and its name is Carboni liver, basically referring to the black staining, black bruising. Carboni liver means black bruise. So it smells like curry. It has a wonderful pink cap that fades away to brown, and the fading begins at the center. So the disc can be brown, whereas the margins of the cap will still remain pink. It has a dingy, as you can see, yellow stipe, but it's often flushed with some pink as well, streaky. Overall, the appearance of this mushroom in terms of its colors is kind of muted. You know, it looks very nice, but it's not bright. And it smells like, doesn't smell like bouillon. It smells spicy, like curry. A very complex odor, unmistakable. You will not confuse this with either Thallax or Polyderosia or anything else. And again, it's only known from Stoke State Forest to New Jersey, as well as a couple of locations in Wisconsin. Hunter collected one of the specimens. On the mow, it's known as species New Jersey 01 for the time being. But like I said, it's going to get its name very soon.
Is that foot staining red?
I'm not sure. Could be a bruise. I can't tell. Could be a transfer from something else. I mean, a horizontal.
Yeah, it looks like it was
vinaceous cut or something like that. Yeah, I don't know what that is. Now, the third bolete comes from a small but very beautiful genus called Calabalatus. And this is Calabalatus rosipes or rosipes. I'm not sure how to pronounce that, referring to the rose-colored stipe. This critter is very similar to Calabalatus inedulus, except that inedulus has a tannish cap, kind of a beige, whereas this critter, which is associated strictly with the hemlock, not with the oak, is grayish brown. It's going to be bitter tasting, but again, it's not very brightly colored, but very beautiful bolete. I really like Calabalatus. It's a very nice, tidy genus.
Is there a species on this one?
Rosipes. Yeah. Roseipes. You can see the cap is sort of grayish brown.
How many species are in Boletus?
Boletus, overall globally, let me check quickly here. If I can upload Index Fungorum, which is not uploading again for some reason.
There used to
Globally,
be
probably, I would say maybe no more than 15. In the USA, we have Rosipes, we have Pecii, we have Inedulus, we have what's that one with the red pores?
Calipus.
No. Calopus is European.
Oh, okay. I was going to ask if that's still, because in the old field guides it used to
Yeah,
appear.
you can forget about that. That's European.
Mhm.
No, it's not responding. What's the name of that? It grows mostly in the southeast in North Carolina and Virginia. The name escapes me. And we have another species that's undescribed. Only one collection exists from upstate New York. Very different DNA. So I would say, 5 species of Calaboletus in Eastern North America so far. Okay, so to finish off, Dave knows the species very well. So when I challenged him with it, he immediately gave me the name. So this is Amanita submaculata, a very young one. This is from section Validae.
The actual color is a little more gray, I would say.
It is gray, right?
Yeah.
But these can be completely white, and they often do have warts on the cap if they're not washed off by the rain, but they're very fragile. Like in all Validae, they fall off quickly, and they tend to be patchy or sometimes even pyramidal. I believe the Stipendus species may stain reddish when you touch it, but you can see the ovoid bulb, no shoulders. Some specimens actually don't even show a distinct bulb. They kind of, enlarges non-specifically toward the bottom. And it smells, if you smell, it has a very fruity smell, but it's very imperceptible. To me, it smells like a minty toothpaste. Some people describe it as floral. The ring is very large. On Rod's website, I think it's called a ball gown Amanita, like, that dress, women's dress with a corset. And, imagine like 19th century, something like that. And very large ring apical, very long and very, very skirt-like. The color of the cap varies. I think we have a collection from the Pine Barrens, which is rare. It's completely brown. So this can come in a variety of colors, but most often white or grayish white or grayish brown white, something like that. Sort of very pallid.
Sometimes it starts out white and then it gets darker as it
Correct.
ages.
So it almost always starts out as white or
Yeah.
grayish white.
And the name submaculata comes from the pattern that sometimes develops on the cap surface. It looks like eyes.
They're like brighter spots, the pigment kind of a mottled appearance, but it comes like in these oval shaped, spots. So sub means under, like, or somewhat like. Maculata means spot, right? Macula lutea, that's a feature in the human retina. Something like that, right? So submaculata. In this case, we don't see the spots.
Yeah,
but once you see the species a few times, you'll be able to distinguish it very, very well morphologically from everything else. I think phylogenetically, this is one of the earlier species in section Validae, basal to the whole section, together with canescence. So it's more ancient, older. And the lack of bright colors that you see often in Validae basically confirms that colors have evolved later on, and earlier Validae are kind of very modest looking. Not much color in them. They're whitish, off-white, mostly. Gray.
Thank you, Igor.
That's all I have to say.
Okay, we have Dave and Hunter. Dave, do you want to go next?
Sure. Let's see. What did I send? There's been a lot of mushrooms to see here. Oh, Rusula dissimulans, right? You know, I've called it dissimulans. I imported this from iNet, so I think it's going to say I'd call it that. But I really should go on to this observation and downgrade my confidence to promising and say that could be Rusula densifolia. So I think this is probably dissimulans. The gills are, I would say, subdistant. These little drops are not latex. It's just some liquid because there had been a lot of rain. So the ground is very damp. So when you slice through the gills, it takes a little while. First, you don't see much of anything. And then after maybe 5 minutes or so, you'll see it starts to get red. So it stains red. And then slowly that red goes to brown and eventually black right at the end. So it's one of these blackening, red and black russulas, they sometimes call them. They turn red and then they turn black. So there's two species. Dissimulans has gills that are described as subdistant to distant, and densifolia has gills that are described as close to crowded. So the difference between close and subdistant is really kind of tricky in my estimation, but I think dissimulans fits this one a little bit better, and it's something I usually find with hemlock.
Okay.
Is that
So
a fetid Rusula?
Excuse me?
A fetid, FOETID?
No.
Oh no,
They're
no,
not.
this is not one of the stinky ones.
No. Okay.
Yeah, this one doesn't have much odor at all, I think. Yeah, it's
Oh,
not slimy either, except if it's, really, really wet outside.
Yeah, one thing I would like to add is that both the simulus and the Densifolia are European species, and their occurrence on this continent has not been confirmed, I think, genetically.
So I probably should have taken home a piece of this and saved it. I think it was pretty well riddled with bugs, actually. Yeah, but this is probably a good one to save some samples and send off for sequencing.
Right. We may have our own species, but it is possible that something like that from Europe occurs here, because we've seen examples of Cortinarius and other gilled mushrooms, mycorrhizal, that are in North America.
So here's another Russula. This is Russula clavipes. This is a large Russula. This cap was maybe 3 to 4 inches wide. And it's one of the shrimp Russulas. So it's sometimes we might say it's in the Zarampolena complex. It stains brown on the stalk. You can see there's a brown stain on the stalk. If you damage the gills, they will also stain brown. It might take a little while, but it smells like shrimp. It's a very good edible. This species is reported to grow under spruce, and that's where I found this one, under one great big old Norway spruce. So at Hickory Run State Park.
So this is also a European species because the author is Velinovsky, and that also came from Europe.
Yeah, so probably I don't know. We ate this one. I probably should have saved some of it. It was in good condition.
So this is like a Zarampolena-like species, or not?
Yes, it's in the Zarampolena complex. Or they used to call it Zarampolena variety clavipes, I think, 25, 30 years ago. It's a mottled cap like you see here. That's part of the description.
Is it brown color because it's
brown, greenish, reddish. You know how russulas are. They can manifest in different color combinations, but kind of mottled, not real bright.
Okay, and the cuticle peeled?
I didn't do that.
Because to me, it gives an appearance like it wouldn't peel at all.
Yeah, it's supposed to be adnate, and it looks like it is.
It's
not
adnate
peeling on its own at all.
Right, it's adnate, and there are no striations or tuberculations on
Right.
the
Yeah.
on the cap.
Yeah.
Beautiful.
hmm
Okay. So you ate this one, you said, right?
Yeah. It was, well, I just put it into a stir fry. There were lots of other things in the stir fry, but you
Okay.
know, it's must have tasted pretty good because the whole meal tasted good.
And it is found only under conifers, right?
Yeah, according to Michael Quebec, it's a spruce a fir associate, I believe.
Got it. Thank
Yeah.
you.
So here's something you might mistake for a Ramaria. Phaeoclavulina flaccida. This was growing in needle litter under Norway spruce, and apparently it's a saprobic species. So what are the differences between this thing and Ramaria? Well, the species name actually indicates one of the main differences. The branches are they're not brittle, they're not like resilient. They're very flimsy, sort of. You can bend them really easily. They bounce right back. Now this young one right here maybe wasn't as flimsy because they're not expanded yet. I got some spores. I didn't actually try to make any measurements, but the spores look like what they're supposed to look like. So one of the descriptions is that sometimes they're comma shaped, which a few of the spores that I saw were in fact comma shaped. So I preserved some of this, and you can see these spores. Some of them actually do look a little bit like commas. So, and it looks like they're probably up to almost 10 microns, and that's pretty much what the expectation is for that species. So I'm pretty sure I got this one right. That's interesting because of the way it looks like a Ramaria. So there's a few other Phaeoclavulina species that they look pretty similar to this one. Some of them have developed stains, but I think that Flaccida is the one that is, as the species name would suggest, kind of flimsy, but it doesn't break easily. It's just easily bent. Let's see, what else do we have here? Oh, right. So this was collected at Hickory Run State Park in Pennsylvania, but this came in at Stokes also the other day, and so there was a little bit of debate about whether or not it was Hohenbiohelia or Crepidotus, and these are easily mistaken for Crepidotus. But these guys have white spore print, which Crepidotus do not have. They have brown spore print. But the coolest thing about these little sort of tiny oysterette type things is microscopic. If you smash mount a little bit of gill, you get lots and lots and lots of these bottle-shaped hymeneal cystidia. So there's a whole bunch of them there. And sometimes there's so many of them that they sort of get in the way of each other, and you can't really see any one or two distinctly. But this photo right here shows a fair number of them. So there's one that I searched around for quite a while before I was able to find one, that looks nice because there's nothing in the way. So there are a lot of them. The little tiny bit of smash mount of gill. And the thing that's kind of interesting to me is that one of the hypotheses about cystidia is that some mycologists believe that they help create space between gills, so that the spores are more effectively come out of the gills, like is what the mushroom wants them to do. So why is that interesting here? Well, because if you go back to the very first photo and you look at, if there's one of them where we can see the gills, I should have tried to get a good close-up on the gills. The gills are very tight on these. They're very, very tight and very, it's not a great photo, but you can see they're very close together, and they're pretty shallow also. Not the best photo, but you get the idea. So
Now this genus has the gelatinous layer. Can you tell that when they're so small? Pull them apart.
It's sort of, yeah, you know what? You sort of see it. especially if you look at the other two that aren't inverted, see where they sort of like go down into the little stipe-like thing. Yeah, you can see it looks a little bit gelatinous. But the main thing is that, to me, is the cystidia. That sort of distinguishes this from other things that look like it, and the white spores. So there's a larger species, Hohenbiohelia petaloides, that grows in clusters right on wood, or sometimes wood like it's on the ground, but it'll be right near wood or buried wood. So
there's a couple other species of smaller Hohenbiohelia, right? That kind of tomentose on the cap.
Yeah, they just look a lot different than these guys. They have like scaly caps. I think there's one that's like bluish or something, grayish sort of. And they're shaped a lot different. Yeah, there are several small species of Hohenbiohelia that form small fruit bodies, but they don't look like Angostata. They look a lot different. So this was on a Wyoming Valley Mushroom Club foray. One of our members spotted this in the woods, and then I saw it, and I thought, well, it really looks like a destroying angel, and the photo doesn't really show the luster of the white color of the cap. Destroying angels have this sort of luster that makes them look a little bit different than, say, the white Amanita brunescens, Amanita aestivalis. The photo didn't really capture that. But why do I put no name on this? Well, we're getting to that. And by the way, you see this snail? This snail was munching on this thing. So this is almost certainly a very poisonous mushroom. So the snail doesn't seem to care. Why is that? Well, who knows? Maybe the effects of the toxin take longer to kick in than the actual lifespan of the snail. I'm not sure. But look at the bottom of the stalk. I did a lot of editing here to be able to see the base of the stalk. There doesn't seem to be a lot of basal structure, although that may have been lost in the soil, because this was fairly well buried and everything was wet. So a lot of stuff was soft and it was hard to get things out of the ground intact. But here's the real kicker right here. There is a drop of KOH on this cap. It's right in the middle of the cap. You can see where there's sort of like, it made a little indentation, just made a little shallow indentation. It didn't turn yellow. So if this is a destroying angel, then it's one of the ones that doesn't turn yellow. So I proposed on Mushroom Observer, what's that name? Amanita elliptosperma. So there are a small number of destroying angels that don't turn yellow when you put KOH on the cap, and that is one of them, or it's a name that's applied to a complex. But I don't know what this is. Is this elliptosperma, or could it be something in Validae like aestivalis? I don't think it's aestivalis. I got spores, and the spores are I think the spores for aestivalis are probably going to be a little bit less elliptic, maybe a little bit more broadly elliptic to subglobose. And these are some of them are broadly elliptic and some of them are ellipsoid. So I think this might be one of these species in the elliptosperma complex. I should have saved it. I didn't save it. I should have dried this out because it seems like a pretty interesting find. So that was my bad. But if anybody has any suggestion here, I would welcome it.
Can we see some pictures again, like this one? Trying to just make out the structure of the bulb. It doesn't look like aestivalis or brunescens, because that one's
it doesn't look like aestivalis or brunescens. I found some photos of elliptosperma online, at least what somebody called elliptosperma, that had this sort of ovoid, like slightly pointed bulb, like you see here. See where it's like sort of ovoid, And so I did find a few photos that look kind of like that. So
Yeah,
I
this
think
is
that's
certainly not the bulb of Estivalis or brunescens, so we can rule
no,
these out.
that's what I would say. I don't think this is in Validae.
Yeah, and overall, this looks like a destroying angel to me. You know, even without looking at the bulb, just you know, the little things that we
Yeah,
usually.
well, see, the thing is, though, when I first saw it, I thought, oh, look at this destroying angel. Let's dig it up and I'll show everybody, you know, the basal structure, you know, the limbate, cup-like sac sort of surrounding the bulb. But none of that was there. So it just kind of threw me. And the shape was kind of weird too, I thought. But a lot of the typical destroying angels, Bisporigera, Mera virosa, they have like more sort of round bulbs on the bottom.
It looks
Right.
like there's like a little bit of red staining on the bulb, or maybe
Dirt, I think.
what's that?
Just dirt, I think. You know, it's dirty. It's just you know, soil. It's
Okay.
just
Is
soiled.
there another photo though? Maybe if we see the other photo though. This photo right here is highly edited just to be able to see the shape. I don't really see.
On the
Yeah,
step, I
no.
think there could be some pinkish staining. You know, that in the middle, that portion of the stipe that's between the cap and the bulb. In the photograph, if you just scroll down, you'll see some pinkish tinges maybe.
Okay, so the bottom part of this photo we'd want to see.
Yeah, the bottom part of this photo. Yeah.
Yeah.
Hello?
Yeah, if it's possible
Yes.
to scroll.
Scroll bottom this way.
Yeah, you see? Yeah. Right there where
Maybe
the
a little
slug is.
bit.
Oh
Yeah, a
yeah.
little bit. Yeah. That might be because of the slug too, you know, no.
A reflection from the slug? Yeah, maybe.
Well, it might be the slug may have climbed up this thing and secreted something.
Maybe.
I think sometimes elliptosperma can have like a little pink tint to the gills, like just a little really light.
We see in some, I think there's one species of destroying angel that has a pinkish gills, and you can see some pinkish tinges here too. Whether this is just an artifact of digital photography, I don't know.
Yeah, I'm not sure. It looked white to me, but there may have been a slight tinge. You know, it was after a foray. There were so many mushrooms I brought home. You know how that is.
Mm-hmm.
Yeah.
Yeah, hardly spent any time looking up what we found at Stokes. I was just busy with my things as usual.
Yeah, there was a fair amount of stuff came in at Stokes actually.
Yeah, I remember. You know, we had a full table.
Yeah. So anyway, that's this thing, and I think elliptosperma sort of so that sort of helps me believe that's what it is. You know, having this little discussion.
Okay.
I would just propose section Phalloideae for this one.
Yeah, I think that's what I'll do. I'll put
Just to be
that
more
as
specific.
promising and override subgenus Amanitina.
Yeah, because Amanitina contains so many things. It's
Yeah, I know. It's just very broad.
it contains
characterization.
everything other than section Amanita. You know, it contains Validae, Amygdala. It contains the bulk of Lepidella, what used to be known as Lepidella. Contains Phalloides, so it contains several sections.
Yeah. Yeah. Yeah, I know.
So
It's a
this is
very
very
general
broad name,
classification.
not very useful.
Yeah. No, it's not very useful. I know. But it was a starting point. And now I will go both to this observation and to the one on INAT, because this was an import from INAT. And I'll make a different proposal. Yeah. And the one thing I don't like about INAT actually is, well, there's a few things I like better about MO, but I like about MO that you can propose a few different things and just give them different levels of confidence, or even the same level of confidence, and the IDs, they all stand. None of them get crossed out. So just because you make an alternative proposal, you're not necessarily saying, oh no, this is not what this is. You know, with identifying fungi, sometimes it comes down to like, you know, well, I think it's two, you know, something among two or three of these names that I'm proposing. And I think that's just a better way to do fungi.
Okay, we just have a few more minutes left. So maybe we can move on to Hunter. You still with us, Hunter?
Yeah, I am. But if Dave has more, I'm okay not sharing too.
No, you go. You go. Go ahead. If you have stuff, you go. Looks like this
Okay.
looks interesting.
So I'm just got to northern Wisconsin today, and these are from today, so I haven't had much time to do much with them. But I think this is Helvella crispa. It has the reticulated stem, but it's very tiny. So it was, I mean, 2 inches tall at the most. Really small. And mostly oak. Is that actually like near some Indian mounds. And yeah, so but I think that's what it is. And I mean, I
Yeah,
had no
yeah.
time.
The size, I think, actually kind of falls right in line. Crispa,
Okay.
Crispa can be pretty small, I think.
Can it also be pretty big? Like, is it?
Yeah, you can get bigger ones also. And I think
Because
they
it's
call this stock fluted. So
okay.
it's like it's got like these longitudinal sort of crevices.
Yeah, so
Yeah.
cool.
Yeah.
It looks exactly like the thing that I called crispa a couple weeks ago.
Yes. Yeah. Okay.
Yeah, so just on micro Quebec real quick, 1 to 4 parentheses 7 centimeters tall, 2 to 6 centimeters wide.
Yeah, there you go. It's a
Okay.
pretty
That's
wide
really
range.
small. Okay. All right. Cool. Yeah, and I some of my pictures didn't really make it, so I'm not sure what actually made it in my email. Sue. Uh-oh.
Did we lose Sue? I know we're getting some weather in North Jersey, so maybe she lost.
Yeah, I lost the Wi-Fi. I'm back. I'm back.
Ah.
Can you see me?
Oh,
I see
I
the
can
knife.
hear you for sure.
Let me try to share again. Yeah, I dropped the sharing.
See, while you're doing that, I'm just going to provide a little more of an answer to your question about the Hohen biohelia, because something just occurred to me. That those fruit bodies are really resilient and rubbery, which I think is probably an indication of the gelatinous substance. Unlike Crepidotus that are crumbly, usually.
Well, that's good distinction.
Everybody can see the knife with the Helvella?
Yes.
Yeah.
Okay. Oh, that's the
So
next
this was a Amanita that I found. It's got some striations. It has some veil remnants. It's very pale, very small. I mean, not like maybe 4 inches tall. Dainty, and I'm not sure what it is.
It's actually Amanita. Oh boy. Do we get a picture of a bulb?
I have
No.
them. I just somehow they didn't get. I had trouble because I was just pasting from my iPhone, and I'm not at home, so I do have them, but I can't get them to you because they're not on the device I'm on right now.
Right. Possibly subluridellus piece. That would be my guess. Just looking at the cap.
Okay. And then I saw other ones that looked very similar, but didn't seem to have the veil remnants. They were maybe like what was it Brandon who showed the was it submacul? No, not submaculata. That little yellow. Oh no, elongata. Yeah, no, it wasn't really like that either.
Can I?
Yeah,
Yeah.
those are a different section.
Yeah,
These-
different
Yeah.
sections.
Yeah,
Me,
these
this-
have true striations. Look at how long the striations are in the upper left.
Yeah, I mean,
And-
you have to
and-
be careful not to confuse this with section vaginata, but the velar deposits on the cap to me suggest section Amanita.
Yeah, I think this is one of these. Nobody knows what they are. section
Multisquamosa or
maybe.
Yeah, there are these
Validae.
panthers and gemate, pantherina and gemate, those two
Yeah.
difficult subsections of section Amanita.
I do remember that the bulb, it didn't have that rolled stalk appearance, and it didn't have the like a chrysoblema look. It was a real bulb. And I'm sorry the picture didn't make it.
But more or less smooth, but maybe with a few patches here and there.
Then probably it's subsection gemate, and this thing probably would not even have a ring.
So it seemed like it might have had one, but when I picked it, it wasn't there.
Yeah, it was
some
probably
of them have
quickly
flimsy
lost.
rings. Some of them have very flimsy rings that don't last very long.
Yeah.
Some people would call this russuloides, maybe, but we don't really know what that means.
So in that case, if the ring, was easily detachable, or however you guys just described that, does that rule out subvolapes and multisquamosa, or?
I think
Yeah.
so.
Not
Those rings tend to stay. I think it's section gemate that either don't have a ring at all, or the ring is quickly lost.
That's usually the case, but sometimes the panthers have partial veils that try to cling to the cap margin, and then they tear apart instead of form an annulus. But usually they form an annulus. They form the inverted funnel annulus. So.
Oh right. Yeah, and I think I had one of those we saw one. We talked about that one last week too.
I think so. Yeah. Well, the main thing is though, you don't see that like that gutter going around the shoulder of the basal bulb that you see on the panthers. These chamate things have more or less a smooth bulb and sometimes if some veil remnants. I'm not even sure if they would be universal veil remnants or partial veil remnants, just get deposited either in the soil near the bulb or on sort of little patches on the base of the stalk. But you don't have that notable structure that you see on the panthers.
Right. Well, Hunter just sent me additional pictures of this Amanita, and the picture that has the bulb does show the color. So I do think this is a panther.
Oh, so it does have a collar on the bottom.
Yeah, it does.
Oh, okay.
Okay, sorry. Yeah, and then it's got like basal mycelium. Do you see that too?
Yeah, I see these rhizomorphal sort of short, threads projecting from the bottom of the tip of the bulb. And this was growing, actually, I see hemlock needles.
Yeah.
So this probably is going to narrow this down to Dave. So what's growing with hemlocks that would have a look like this? Praecox?
Oh, Praecox doesn't have the well-defined gutter going around the.
Okay, then Albocreata?
Albocreata does, but that one's usually paler and it just has like a yellow disc.
Okay. Now the other picture that Hunter sent me of the whole mushroom. Let me just blow it up to make sure that
Albocreata has no partial veil at all.
Well, this one does have a partial veil. There's no indication of.
It's probably Praecox.
Yeah.
Yeah, Praecox. Lamoureux authored the species Praecox. It's provisional, I believe, but he says there's no partial veil. I do wonder about some of these section Amanita things that supposedly have no partial veil. I think sometimes they might have like a rudimentary, very, very thin membrane covering the hymenium that breaks apart really early. I've seen it on albocreatia, and I'm almost certain they were albocreatia. I've got photos of it online. But Praecox is supposed to be ringless. Albocreatia is supposed to be ringless. Those are panthers. So, or no, I'm sorry, Praecox is. I asked Rod once if he thought Praecox was a panther, and he was kind of non-committal about the answer. I think he said something like maybe.
Yeah, but the bottom line is, regardless of whether you can confidently assign your Amanita to that subsection or that subsection, if it actually turns out to be phylogenetically supported, deciphering these things is a non-trivial problem. You have to use DNA.
Oh yeah, absolutely.
And combine that with the morphology, you have to do a lot of sequencing and collecting, just to see if you can see anything in terms of morphological traits. You can actually separate them in the field eventually. These are a big pain in the neck. These sections of Amanita are just very difficult.
Yeah, well, that's why I said a little while ago that probably nobody knows what this is at this point.
Right.
In time.
Section Amanita series Chamata. Is that where we left it at?
Pantherinae, probably.
Yeah, because of the basal structure.
Yeah.
Mm-hmm.
I'll forward both Dave and Brandon Hunter's photographs so you can take a look at those things.
Are we saying Pantherina is a species or a subsection?
No, there's an Amanita pantherina from Europe, and that's the type species for the subsection, I believe. So this is a panther-like or pantherinoid Amanita, which means that it comes from series or subsection Pantherinae. Pantherinae.
Yeah, they're not black at all, though. Igor, you could put those in the chat, maybe. I'm so sorry that I can't.
Well, I cannot put photographs in the chat because they don't have links to them. They came as an attachment, and I don't think it's going to work with the chat.
Oh, okay. I thought you could copy paste them, but maybe not. Okay. So this is, I'm thinking, a marasmus of some type. It was really pretty. And if you like blow up on the gills, the gill shot, and see how pink it is in the center, that was really pretty, and it was much more vibrant than the picture shows. These were really small, like the biggest one maybe a little bigger, maybe dime size.
So it's more pink than orange, you think, in the middle?
Yes, they weren't orange, they were pink.
Yeah, because there's an orange one and there's a pink one. The orange one is Siccus, and the pink one is, I think, Pulcheripes.
Okay, this must be Pulcheripes then. I also saw the orange one as well, and I don't think the picture made it into my email, but yeah, I saw those too. And those, when I see them, are usually a lot bigger, but they were also really small today too, the orange ones. But yeah, this was really pretty, and the stem was so thin, I couldn't believe it, but sturdy-ish.
And I don't know much about the pinkish ones, but I've tried to ID some of the orange ones, and Siccus is there's a few things that look exactly like Siccus, the orange ones. So I think that one's kind of a complex or group or something.
Do they have other species, Siccus-like species on Micro-Quebec?
Not sure.
Or did you just find like things on your own that you thought were different? Because it's very likely a species complex.
Well, let me check on Mycoquebec a couple places. Yeah, this isn't something that I was like, oh, I think this is a species complex. It's, I saw somewhere that there are a bunch of things that are falling under the name Siccus that aren't actually Siccus.
Oh, okay. Yeah, I don't think I've run into that.
I believe it because this one I found seemed quite different from what I know of as Siccus in Illinois. This one that I tried to include, I'm actually sending it to poor Igor again.
Well,
Michael-
honestly, Siccus and Pulcheripes don't have so much white on them as these.
Michael Quebec has Marasmus bellipes.
Oh, right. I forgot about that one. Right.
Maybe it's that. That has pink on it in the middle.
Okay.
That's like pine.
And it's dark. I don't know what it grows with.
Bellipes.
Does that one have more of a white margin like this? Because I think Pulcheripes is a little bit more completely pigmented.
That's what it looks like a mushroom expert, much more pink.
Yeah. Okay, so Bellipes, that sounds good. Let's have a look at the photos. I'm going to look.
Bellipes.
So Brandon, those are the ones that are confused with Siccus?
Yep.
Oh, okay.
Yeah, because I looked into this last summer, and I looked into some sequences or some Marasmus siccus things that were sequenced, and came back as matches for siccus that look like your thing. They look completely different from what I think siccus would look like, and then some of those kind of textbook siccus. You see it in the field guide. Oh yeah, it looks exactly like that thing. They come back as other sequences. So
Sue, can you open one more email that I just sent you?
Yes.
If it comes.
If it
It's-
comes, yes.
Yeah, if it comes. Okay.
Into the chat, I just pasted the Mycoquebec link for Marasmus Bellipes, and
Oh, here it is.
I think that's what those are.
Okay, and so then these were orange though, and
Oh, sorry. Sorry.
and then I've got the
Well, that's just a look.
No, it's a different one. She's done a different thing.
Yeah.
Different.
We're on to
yes.
different ones.
So this is a different one, and it's orange, and so
Sullivan.
I mean I was gonna call it Siccus or Siccus or whatever.
Does everybody think this is Siccus?
That's what I'd call it. It's very orange.
Yeah, it might be. It's more Sullivan. It's not.
Okay, so it's not a mystery, and that's good. Yeah, these were pretty. It's very wet up here, but it's a little bit early in the season, so we'll see what I find. I did find some other cool things, but I'll send them next week because
Okay.
my email didn't come together too well.
Yeah, I was wondering what happened to you because I'm so used to seeing yours amongst the first.
Yeah, I've been I don't know, busy, but I've been, yeah, it was, things dried up at home and it's been so hot. Like you guys have had the heat too now, right? The 90.
Yes, we're 90 degrees plus, yeah.
Yeah, I think we've had it for like 2 weeks straight and we were getting rain, but then, yeah, it just kind of died out. So I was waiting to find stuff today for today, but it's just too much in the day to make happen. But I'll get them organized next week and share them with you guys.
Okay, well, thank you, everyone.
Thanks,
I hope to see
everyone.
everybody next week.
Yeah.
All right,
And for
thank
those
you,
of you who came
Sue.
out before, right?
Yeah, thanks, Sue.
All right. Have a good night, everyone.
Good night.
Right.
Thanks.
Yeah, good night. I'm going to sign off. Oh, there's Phaeolus.